Okay, hello everyone, welcome back. So far we have learned about lipids and carbohydrates. Now we're going to go on to our next macromolecule, which is proteins. This one's going to be a lot of information. Proteins are super important and there's a lot to know about them. Okay, so proteins. So the monomer for proteins are going to be amino acids. When you get a lot of these amino acids linked together, you will make proteins. So the polymer is going to be called proteins. So all amino acids have this same basic structure, and it has an amine group, so that gives rise to the amino part of its name, and it has a carboxyl group, so carboxylic acid, which gives rise to the acid in amino acid. It also has this carbon and this hydrogen. So amino acids are very polar. If you remember, the amine functional group is polar, as well as this carboxyl functional group, so both of these are polar. The difference between all these amino acids is going to be the side chain. This is sometimes referred to as the R group. So we have 20 different amino acids, and this structure up here is the same for all those amino acids. The difference is going to be this side chain. So amino acids so here you have one amino acid, here you have another amino acid, and the difference in the side chain is just depicted here by this difference in shape. We have a triangle and a square. These two amino acids can link together through a dehydration reaction. So this OH group and this hydrogen will form a water molecule, and these two can now link. These will be linked by a peptide bond. So the bond that joins adjacent amino acids is called a peptide bond. The resulting long chain of amino acids is called a polypeptide. So if we had a bunch of these amino acids linked up, it's a polypeptide, and eventually it gives rise to a protein. So a protein is a polymer of amino acid monomers. We already discussed that. All proteins are made by stringing together 20 amino acids. These are the 20 amino acids. So I picked this image because here you can see that all amino acids share this amine group, this carboxyl group, this carbon, this hydrogen, and the only thing that is going to be different are these R side chains, and those are highlighted here in yellow, blue, purple, and green. You can see that these can be very simple. So our most simple amino acid will be glycine, which is shown here. You just have a hydrogen as the R group, or it can be very complex. So our bulkiest amino acid is tryptophan. You have two ring groups here. Let's see what else I can highlight about these amino acids. You definitely don't have to memorize these amino acids. It's just kind of good to appreciate the differences. So like I said, glycine is your most simplest amino acid, just as hydrogen. It's the smallest amino acid, and it's going to be the most flexible because it doesn't have this bulky R group. Proline is considered your least flexible, and it's because it builds back up onto each other like this. It's see how these R groups don't build back onto the amine group? This R group is going to build back onto this amine group here. So it's your least flexible amino acid. You have acidic amino acids, which are going to be your, here we go. Your acidic amino acids have acid in the name, and they're both going to have this extra carboxylic acid or carboxyl functional group. And just so you guys know, all these amino acids have these abbreviations, these three-letter abbreviations. Once again, you don't have to memorize this, but sometimes you will see them written out as glycine GLY, alanine ALA, things like that. And essentially, you can bond these amino acids to make different proteins. So you can have like glycine, alanine, lysine, histidine, and that makes a protein. Or you can have repeats like glycine, 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 leucine, 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 serine, tyrosine, glutamine. That makes a different amino acid. So as you can imagine, there are very different combinations that could arise from pairing these differently. So each amino acid has a unique side chain, which gives that amino acid its special chemical properties. It also gives it a different structure, and if you remember, structure dictates function. And that is shown here. So here you can see how these side chains will give rise to very different structures. And like I said, tryptophan is very bulky, and you can see that here in its structure compared to glycine, which is not very bulky. Okay, another thing I want to point out is that these protein structures will have different primary, secondary, tertiary, and quaternary structures. So primary structure is basically just going to be, you have your, this one's actually not that nice. I'm going to use this top image. But primary structure is just these, they're just being bonded, these amino acids by peptide bonds. So here you have your one amino acid, here you have your other amino acid. They come together in a dehydration reaction to build a dipeptide or dipeptide. And as you can see here, typically your R chains are going to alternate directions. So you have your R chain on the top, R chain on the bottom, R chain on top, R chain on bottom, etc. You have an amino end, which is your N-terminal end. So here's your amino group. And you have your carboxyl end, which is your C-terminal end, or C-terminus is what we refer to it as. And these peptide bonds, they behave a lot like a double bond in the sense that they cannot rotate. So they're going to be fixed. So you can have rotation here, fixed, rotation, rotation. Then you would have a peptide bond, which is fixed, rotation, rotation, fixed. And this is important when we go on to secondary structure. So this here is a secondary structure. It's also shown here. So essentially in your secondary structure, you have all these protein or amino acids that link to each other through peptide bonds. And then they're connected with each other by hydrogen bonds. And this ribbon isn't really there. It's just to help us illustrate how they will form these alpha helix like structures. And typically what you find in these alpha helix structures, the amino acids that will form these alpha helix like structures will be smaller amino acids. Your bulkier ones tend to form a different type of secondary structure, which is called a beta sheets or beta pleated sheets. Or, so these are beta strands that will be connected to each other by hydrogen bonds to make these beta sheets. And you do have proteins that can, I guess you do have proteins in nature that only have the secondary structure. And an example of that would be fibrous proteins such as hair. So hair only exists in the secondary structure. And essentially when you have curly hair, it kind of has this helix-like structure, right? And then when you want to straighten it, you have to break these bonds. So a perm is basically breaking disulfide bonds, and that's why it stinks, because sulfur has that egg-like smell. So you're basically breaking these bonds to straighten it out, to make it straight, lose this helix-like structure. And that's how a perm would work. And this is called, this phenomenon is called denaturation. So you could expose a protein to high heat or different pH values to make it denatured, and it basically will lose its shape and make it straight because these bonds are breaking. Another way to think about breaking hydrogen bonds is by boiling water. You boil water, these hydrogen bonds are going to break. So exposure to high heat will break bonds as well. And then let's see what else I want to say about this. Okay, so then we also have a tetrary structure, which is shown here. I believe this is going to be myoglobin. And this is an image here of a tetrary structure. So you have your alpha helixes shown here in these red ribbons. And then as you can see, these are connected by these bends. These bends will typically be proline. So proline, like I said, is it's not a flexible amino acid, so it tends to be seen in these bends typically. And then you would have your quaternary structure, and it's basically bringing all these subunits together to form a complex protein. So an example of this would be hemoglobin. So you basically have 2 alpha subunits, which are shown here in purple, and 2 beta subunits, which are shown here in this teal color. They come together to form this complex quaternary structure, or in this case, hemoglobin. And so, like I said, amino acids are protein monomers. They all have a carboxyl group and an amine group and a side chain, which we refer to as the R group. So here you have your amine group. So your amine group is a nitrogen and 2 hydrogens. You have your R group. So here we just have the carbon with the hydrogen, and then you would connect your R group to it, to this backbone. And then you have your carboxylic acid or carboxyl group, which is your carbon, 2 oxygens, and a hydrogen. So you combine these together, which is shown here. This is your amine group, your CH, your carboxyl group, and here is your R group for alanine. Alanine is going to be a carbon and 3 hydrogens. So if we go back here, alanine is a carbon with 3 hydrogens, yes. Okay, so that is how you would build an amino acid. Each of the 20 different amino acids have a different side chain. And you can look at these in your lab manual on page 39 if you would like, or you can use these images here to look at them. So that is all I had for this. There was one more thing I actually wanted to talk about. I wanted to kind of illustrate the importance of these amino acids. So tryptophan, where's tryptophan? Tryptophan, for example, is used as a precursor in serotonin. I wish I would have put a slide of this, but I totally forgot. But I'll add it in later, but tryptophan is a precursor to serotonin. So if you saw serotonin, it has the same structure here. And serotonin is an important neurotransmitter, and it's basically known as your mood neurotransmitter. And then we have tyrosine. Where is tyrosine? Tyrosine, which is here. This is an important amino acid and precursor to both dopamine and epinephrine. So dopamine is an important neurotransmitter that is involved in our reward system. So it's commonly, dopamine is known to be affected in addiction. It's also affected in neurodegenerative disorders such as Parkinson's disease. Tyrosine is also an important precursor to epinephrine. So epinephrine is involved in our fight or flight response, and you might know epinephrine as adrenaline. So it's just kind of important to see how these amino acids are involved in very complex behaviors. So next we're gonna continue with enzymes, and I will post another PowerPoint for that activity, okay?